All right, everybody, hello. So, you are in the midst of the first episode of Tissue Talks, where I talk extensively about a plant species. Now, today's going to be special. We're going to talk about a relatively obscure native orchid. It's only a few inches tall. It has a single leaf and a single flower, and it grows out of a creeping rhizome in sphagnum bogs and fens and other wetlands. It's Pogonia ophioglossoides, also referred to as the rose pogonia. Now, there are a number of bog orchids that are particularly similar in appearance. They're purple, they have some yellow to them, and they kind of have this sprawly, spidery sort of appearance. There's a number of purple platanthera, for example, that are found primarily in wetlands and bogs. There's Calopogon tuberosus, there's Erethusa bulbosa, there's Calypso bulbosa, there's Pogonia ophioglossoides. Now, really, Jesus fuck. Um, now, bog orchids sometimes rely on a strategy where they mimic a rewarding purple flower. There are a few purplish bog flowers that are similar enough in appearance that provide nectar, a reward, if you will, to their pollinators. And so orchids sometimes rely on deception to get pollinated. Orchids are interesting because, in many senses, they are extreme parasites. Not only have orchids convinced us humans to fawn over them because of their beauty and mysteriousness, they require, because they don't bother to feed their own young, a fungal host. They basically trick with like a chemical coating on their seed coat. They trick some fungus into inserting nutrients into the embryo within the seed to the point where that embryo can erupt out of that seed into an undifferentiated mass of cells known as a protoform that will shortly, or maybe after 10, 20 years, sprout and become a mature orchid plant. This is one of the reasons why it's a bad idea to poach orchids if the concern is to conserve them. You can very easily remove a population by digging up even a small proportion of mature plants. Now, that aside, these plants also parasitize, in a way, a number of insect species. Some orchids have a thing called pseudocopulation, where the flower looks and smells like the female of a wasp or a bee or whatever else. And the poor insect is duped into essentially dry humping this flower until the pollinium, it's like a big mass, a wad of pollen. It's analogous to a spermatophore in an animal, like a big bundle that holds all the sperm inside of it instead of a free flowing ejaculate. We need to stay on topic. The thing is that these plants do things in a really effective manner. And this has led to speciation. Now, I would argue that the degree of speciosis, the number of species in a taxon, is not necessarily the best way to measure its success. And what I mean by that is just because something has a whole fuck ton of species as a result of just various speciation events doesn't necessarily mean that this group is the most successful group. Do orchids make up that much plant biomass on the planet? Just because there are, 38,000 species of them, does that mean that the vast majority of plant biomass is from an orchid? No, it doesn't. Now, aside from that, something you have to recognize is that their particular lifestyle lends itself to specialization. And when you head towards specialization, you really ramp up speciation. That is why orchids are so speciose. Additionally, we came in and decided to do all sorts of, you know, hybridization, whatever. Now, even though that is the case, there are groups of plants that have a more profound impact on our lives. I mean, just don't forget, we are the ones that are defining what a species is and then counting how many things in a category fit that. It's really not that strict. The reason that this is important, people, is because when you have something like us defining species based on levels of divergence in some character, because that's essentially what we do. Even when we're doing molecular data, what we're basically doing is saying, okay, these several taxa look very similar to each other. And as we take a look at that DNA, the DNA of the more similar looking species tends to itself look more similar. And that indicates that, yes, the divergence of characters can be measured, but it doesn't necessarily, strictly speaking, mean there's a hard line where one thing becomes another species, as opposed to just simply a variant of an existing species. One of the issues that we run into is we tend to define species as far as their variability in terms of phenological things like their coloration and things like that, without consideration for metabolic profiles and maybe, response to oxidative stress and other things. I just think that we have to be careful saying that just because a group is the most species rich, it must therefore be the most successful, because one, species richness is entirely a function of how we as human beings decide that these things are separate from each other, but also the role they play in the ecosystem is a far greater indicator of their success, or maybe the role that they play in our lives. Now, sure, orchids are successful in one regard because they have really captivated us. However, things like the Poaceae, the grass family, the family that has a shit ton of edible things that feed like the world half its fucking calories, or the Solanaceae because of the goddamn potato. Okay, I'm just saying, there are not nearly as many solanaceous plants as the Orchidaceae, but remember how many people died in Ireland because their main food source was destroyed by a fungus? Okay. Remember that whole, potato blight? It's just something that you have to. No, sorry. I think potato blight is classified as something else now. No, it's not a fungus. It's that like Hoana flagellating type thing. It's is it a protist? You know what? Let me figure this out. Google, um, boom, potato blight, Ireland. Ba-ba-ba-ba-dum. Phytophthora infestans. What the fuck is Phytophthora infestans, bitches? Okay. Jesus Christ. All right, I'm gonna scream. Oh, it's an oomycete. Okay, yes, that's correct. That's right. That's what. All right, now I remember. All right, so oomycetes are like these, like it's like a weird class of organism. Um, it's it's a eukaryote, but it's like its own class of or phylum, sorry, of eukaryotes. So it's not a protist or a rotifer or anything like that. It's just its own thing. Hang on, I'm gonna get a beverage. I'll be right back. So, okay. Okay, cover. You and I are going to check. All right. Da-da-da. All right, people. Ugh. I need to stretch real quickly. Okay. Oh, fuck. That's better. Okay. So, anyway, what I'm saying is, it is cool and interesting to have a group that is incredibly species diverse. And the reason I say that is because I do like to think of things in terms of species, but my species concept is a little bit different. It basically, hi Bubber, incorporates the general gist of, it's a group of distinctive organisms, distinct organisms that have like characteristics that diverge them from others. I can't remember. I have it in a video. I have it written down. I'll figure it out later. But anyway. And the interesting thing about like the whole species concept with humans, like, let us just talk a little bit, just for a few minutes, about certain species and certain groups of species. The thing is, we are oftentimes defining a species variability based on physical characters. So, what I mean by that is we have a species of Lepidopteran actis luna. I have to try and convince people it's variable to a great degree It is fairly variable in coloration and things like that, but they're also highly variable in metabolic phenotypes, as well as the way they kind of hedge their bets at the end of or at the beginning of the season. Something I'll notice is if I have like a female Luna emerge like late April or something, her eggs at 70 degrees are going to take 14 days to hatch. But a female who comes out 2 weeks later, her eggs at 70 degrees take 11 days to hatch. And I suspect that's because the female who comes out a little earlier is going to have a harder time, like getting those eggs on a big enough hickory leaf or something like that. Because the leaves aren't as big, 2 weeks earlier in the season. But anyway, there's quite a bit of variability in the way that Lunas approach things, if you will. But this isn't about Lunas, so we're gonna just worry about Lunas later. But in the plant world, there are species of ferns like Phlebodium aureum that are highly variable in their physical character, such as the shape of the frond and the coloration, and some of the tightness of the rhizome, like how far apart are the new fronds. Is the rhizome kind of curling in on itself, or is it going every which way? It's variable in those characters. And what you have to understand is that, and then there are ferns like some of the Thelipteris, where it's almost impossible to tell the different species apart, or the fucking willows, for example, where you have to identify a willow accurately, you have to watch its characteristics throughout the entire season. You need to know if it's precocious, serotinous, or coatinous. You have to know things about how the floral buds are arranged. You have to know, the texture of the bark and the wood. Okay, and those are two different things. There are willows that have none of the native willows, to my knowledge, have very deeply furrowed inner wood. But a lot of the native willows have very deeply furrowed and funny notchy bark. That's a different thing. Okay. And that's important to understand the difference. Now, Salus cinerea is a horrible fucking invasive here that is extremely variable in certain physical characters, such as the leaf size, shape, and texture, the arrangement of the leaves on the stem, things like that. Now, the telltale sign it's scenario, though, is the bark itself is this dull gray and it's perfectly smooth, but the wood underneath has like this really stiff, veiny, gross texture. If you rub your hand up, and this is gonna get flat. I need to be careful with my hand gestures. I I don't even know if Emily's in the house. Emily! Okay, thank goodness. Oh my fuck. Hang on. Okay. Let me just see something. Okay. Sorry, I was just checking on something. Anyway, so what I'm saying is that some of those, physical, outer characteristics are interesting, but I also want to bring up that there's, you know, metabolic variability in some species that's extreme. Like think fucking spongy moth Lamantria dispar, where they will eat just about anything. I mean, there are a few things they won't really bother. They don't really bother tulip tree. They don't really bother most of the willow and poplar species unless they're really densely unless it's an outbreak. Um, hang on a sec. Hang on, people. Oh, Jesus. Okay, here. All right, anyway. There. So, there's also, like plants that have different strategies. So, you have plants like Oenithera biennis. Now, Oenithera biennis, I only really ever see in frequently disturbed, like dry, sunny areas because it doesn't respond all that well to competition from other plants. And of course, if the ground is always being fucking disturbed, it's like the fucking intermediate disturbance hypothesis, people. But anyway, um, the Oenithera doesn't like to share resources with other species. So, of course, like when the University of Rhode Island many years back, like was dumping just this fresh dirt outside of like this fucking construction zone that had the like it was near the pharmacy garden and whatnot. A couple of weeks later, the place was just crawling with fucking Oenithera biennis, the fucking common evening primrose. And so this plant, spends the first year building up resources, and then the next year it just puts all that energy into blooming a fuck ton, making a shit ton of seeds, dying, and then the seeds just spread. It's, a plant that's very much into early successional stages, a recent disturbance to an area. They like to be there when that happens. And that's good. You need species that do that because without them being there, you can't get the other species. Okay. You understand that, right? So it's good that you have these weedy things. If you see like a first year Oenithera biennis, it looks like just a random fucking weed in the middle of the ground. And most people will be like, okay, I'm just going to get rid of this. But no, this is a plant that is not only native, a good food resources for a large number of organisms, including yourself. The plant is edible in its entirety, but also common evening primrose is one of those plants that facilitates the arrival of other species to an area. Okay, that's what it's good for, and you need that in an ecosystem. Okay, so that's why it's important to appreciate those native weedy things. Anyway, um now there are some plants that are kind of pain in the ass, like weedy things that love wet areas, like Anoclea sensibilis, Pogonia ophioglossoides, and several other things. Okay, now we're going to talk quite a bit about Pogonia ophioglossoides, okay? I promise. We're laying a couple of foundational things first, but we're getting close to being ready to talk about Pogonia ophioglossoides. Anywho, so orchids are successful in a certain sense, yes. They've kind of mastered the art of taking advantage of just about everything well enough to the point where they're parasitic, but not to the extreme where they are not tolerated. And what I mean by that is, you know, most orchids have very low pollination efficiencies and things like that, which is, by the way, why they evolved to live for hundreds of years. So, plants like Cypripedium macaulay, it's a native orchid here. The plant not only takes well over a decade to go from seed to flowering plant, it'll live over 125 years, by the way, but it takes more than a decade to reach sexual maturity. It might, if conditions are not prime for it, wait until you skip a year of developing. They can do this up to 7 years in a row, and they just kind of have a mind of their own. Honestly, if I'm honest about most of the cypripediums, like I've had cypripedium seedlings that I've planted, right, that have then like not come up, but then if I dig them up, they're not dead. They just didn't bother to grow. And there's like, whatever, okay, fine, you're still alive. So, I have the understanding that, like, yeah, cypripediums sometimes don't like to, you know, they are very sensitive to a lot of things, actually. Um, but they can afford to be because they have this weird strategy. It's convoluted as fuck, but their weird strategy is to be very old, very slow developing, have a very, very low pollination efficiency. But when a seed pod is produced, tens of thousands of nearly microscopic seeds that are barely differentiated embryos go into the world. There's bound to be just that right little spot, the perfect little moist nook or cranny where the, fucking fungus that will facilitate this species germination happens to be, and the plant is able to establish itself. It's a numbers game. Instead of deciding to make a couple of hundred modestly sized seeds that have a food reserve in them. But, then you have to worry about dispersal mechanisms and things like that because now the seed is too heavy to be carried by the fucking air. But anyway, instead of doing a normal plant thing, orchids just say, fuck it, we throw caution to the wind. We're just going to make a shit ton of babies. You throw enough shit at the wall, some of it's going to stick sort of mentality, not literal mentality, obviously, but that's the sort of strategy that the orchidaceae employ. You have to remember that, okay? Now, in any case, the orchidaceae is represented by a great number of species that aren't particularly, for the most part, I should mention, important for biomass representation per se, or perhaps pollinator food resources or things like that. Maybe pioneer species like Oenithra biennis would be. But they have their own little just way of being that makes them just perfect at like somehow managing to skirt along. Whatever, it's fine. Now, there are some some orchids that are more notable than others. Okay. Now, we'll quickly talk about a few. Obviously, vanilla planifolia. It is one of the only orchids that is farmed for culinary purposes. Okay. It's a very important spice. There are some tuberous orchids that are eaten in very localized parts of the world. So there are some orchid species. You can also eat the flowers of certain species without ill effect. I mean, they're not particularly nutritious, but they're not going to kill you if you eat them. But for the most part, there aren't that many orchid species that we rely on for like food necessarily. Um, but where's it going with that? Hang on, I need some water. We do need to quickly talk about Phalaenopsis and Cattleya. Um, basically, in a nutshell, Phalaenopsis and Cattleya have kind of become model orchids or even model organisms for certain plant breeding and plant tissue culture techniques. And because of that, by the way, you have this just interesting contribution to our modern understanding of plants as a whole because of work done in the 40s on a couple of groups of orchids. Okay. Um, Now, this has been instrumental in informing some of like our understanding of what's it called monocot differentiation development yada yada. You know, orchids kind of take being a monocot to like an extreme. And the thing about monocots is they are kind of the evolutionary newcomers, and they are just more metabolically efficient at doing a lot of things. And that is why, by the way, a great number of crops that a huge portion of the planet relies on, rice and wheat and rye and sorghum and barley, corn. Do we say wheat? Do we say rice? We say yeah. Um, Jesus Christ, we rely on a lot of monocots. You know, poaceae, the grasses, those are monocots, by the way. Okay, and part of that is because there are monocots like pineapple. It's a bromeliad. Um, Things that just utilize water so efficiently, like coconut doesn't have access to a lot of fresh water in many areas where it grows, but it manages to do just fine. It's a monocot. Banana's a monocot. You know, a lot of monocots are important to humanity. Okay. Hang on. Who is messaging me? Okay. Nice. What is? Oh, who would do? Okay, I'll worry about that later. Anyway, so orchids have been helpful in our understanding of some of the inner workings of monocots, um, as well as some things like the aeroids. I'm not the biggest fan of aeroids. I mean, I don't mind them, but I'm not obsessed with aeroids. But they're interesting. They're monocots. They do their monocot things. We also have things like what was it? The lady slippers. In some regards, are pretty important in like local lore and things like that. And there's all sorts of just other stuff about some of these species of orchids that are unique. So we'll kind of shift now into Pogonia ophioglossoides, right? That's kind of why we're here. And so Pogonia ophioglossoides is a small orchid that likes really bright, wet areas. Doesn't seem to care too much about the pH, but you're only going to find it in areas that are pretty much inundated with water all the time. They like wet feet, okay? I know of a few populations that I like to go visit soon, but one thing that I've had curiosities about this species is how to propagate it. Now, something about Pogonia ophioglossoides is that it is very easy to propagate. Just not in the way I would prefer. Now, you can take any like small rhizome fragment with an attached root and shoot bud from a Pogonia and put it in some wet peat perlite and just keep it flooded with some sphagnum as well, and it'll quickly develop into this colony of clonal Pogonias. That's the way they spread through the ecosystem. But they also generate seeds. However, there is just I don't know how the hell to fucking germinate these seeds. Okay? And that's why we're here today. That's what we're here for. Okay? It's going to get a little weird. All right? I am going to talk about a series of just fun little experimental things that I did, and then my future experimental plans to try and get the seeds of Pogonia ophioglossoides to germinate. Now, there is an article by a person from, I can't remember all the details. There's an article that was a thesis defense of somebody who did get Pogonia ophioglossoides seeds to germinate on like a Knudsen C medium or something of that sort. But I haven't been able to replicate that protocol. However, I do wonder. I think I made a mistake when I ran that protocol. So I think I am going to rerun that protocol to rule out the possibility that maybe the last time the protocol didn't work when I replicated it was because I just fucked it up somehow. Okay. But from what I can remember, I had made the medium the way that he had described in the just in the thesis or whatever. Let me try to find it because A, I want to read it again, and B, I want to give the person proper credit. One sec. Bear with me. Okay. Hang on. What if? I don't know. What the fuck? Okay. Yeah, I know. I apologize in advance. All right, I will find it. Where the fuck is Google Scholar? All right, you know what? No, I don't. Oh, yes, here we go. Okay. Oh, yay. Wait a minute. I need to see. Hang on. Okay, I need to, you know what? All right. I'm gonna, oh, somebody's here. Hello. Okay. So, oh, somebody left. All right. I'm gonna cap it here for just a, wait, I wonder if I can pause. How do I pause? What is this? What am I clicking? Shit. Am I able to pause my hang on. Yeah, well, all right, we're just gonna close out for a minute.